Hi, I am Mikele Milia, a PhD student in the Department of Information Engineering at the University of Padua. And today I will talk about our published hybrid multi-agent spatio-temporal model, and in particular on how to fed it in order to simulate the tumor microenvironment. The purpose of our work was to model and simulate the tumor microenvironment, also known as TME. When we talk about TME, we refer to the cellular and extracellular environment around which the tumor develops in space and time. And we know that such environment consists of blood vessels carrying nutrients, innate and adaptive immune system cells, tumor cells, and cancer-associated fibroblasts. Since the strong interactions that occur between the cell types leads TME evolution, its analysis can be a key point to understand disease progression. In particular, our framework can be seen as a stack of cooperating layers. We have the cell agent layer, where each type of agent has different possible actions, nutrient consumption, and sends different molecules in the environment. The signaling molecule layers that keep track of molecules like cytokines in the environment. The nutrient layers, where diffusion is modeled through partial differential equations and their concentration are used to regulate aspects like tumor proliferation and survival. Among the different layers discussed above, the first two blocks compose the agent-based model, while the last block composed the PD-based model that, once coupled, leads to our hybrid model must. Focusing on the agent-based model, we modeled several agents, and in particular, cancer agents may acquire several properties, like expressing immunoinhibitory molecules, PD-L1-like on their surface, thus acquiring the ability to escape immune system attack, and we refer to that property as immunogenicity, mutate and acquire or lose antigens, and we refer to that property as antigenicity, evade even immune system by locally releasing signaling molecule, and we refer to that property as adjuvancicity. On the other hand, for the immune system cells, were modeled the natural killer cells that can perform fast immune reaction and releasing specific stress molecule in the environment, the antigen-presenting cells that digest non-endogenous cells and present their antigens to the immune system, releasing molecules that initiate the inflammation, the CD8 plus that, differently from the natural killer, recognize and kill only antigen specific target, the Treg that assist immune system cells and act as support agents promoting recruitment of other agents, the CAF that inhibit immune cell recruitment and promote tumor growth. Focusing now on the PD-based model, we know that nutrients are diffused from their search vessels within the environment, and the nutrient concentration in a certain in a certain position of the environment is computed by solving the following equation at the steady state, where the parameter dc is the nutrient diffusion coefficient, the parameter I, the xyt, and ki are the spatial distribution at time t of the immune system agents and their corresponding consumption rates, and td, xyt, and kt are the spatial distribution at time t of cancer-related agents and their corresponding consumption rates. So, to sum up, the algorithm behind must work as follows. After a random initialization of the cells in the environment and until the simulation runs out of time, we update the nutrients concentrations through partial differential equations, and for each agent or cell in the grid, we stochastically sample and perform an action, while updating the communication molecules in a neighborhood whenever possible. So now the question now is, how can we simulate a specific TME? Well, in order to simulate a specific TME, we thought of how to inform master, and the idea was to use omics data to inform our simulation. And in particular, we focused on DNA, bulk, and single-cell RNA sequencing data. In this way, we are able to infer cell fractions of the tumoral mass, as well as cancer agent properties like the one discussed before, so the antigenicity, the immunogenicity, and the adjuvancicity, that, for sake of clarity, are respectively the ability of tumor to mutate, acquire, or lose antigens, the ability to express immunoinhibitory molecules on the surface of the cancer cell, and the ability to locally repel immune system cells by releasing signaling molecules. As a real case scenario, we consider the colorectal cancer. It is known that colorectal cancer tissues can be characterized according to their gene expression profile into 4 consensus molecular subtypes, also known as CNSs, with distinctive biological features that are from left to right: immune system infiltration and tumor cell hypermutation for CMS1, WNT and MYC activation for CMS2, strong metabolic dysregulation for CMS3, and the high presence of cancer-associated fibroblasts for CMS4. Now, depending on the available data, we estimate different properties. In particular, for the estimation of the antigenicity, we leveraged on the DNA sequencing data, considering the number of mutations present in the tissue. Then, depending on the data availability, we can estimate through single-cell RNA sequencing data or bulk RNA sequencing data the cell fraction and the adjuvancicity, take advantage of techniques such as deconvolution to infer tissue composition while using bulk RNA sequencing data, or clustering-based approach combined with cell type annotation while using single-cell RNA sequencing data. In the same way, we can estimate through single-cell RNA sequencing data or bulk RNA sequencing data the average gene expression of immunoinhibitory molecules PD-L1-like at the level of cancer cells or tissue, respectively. And we also take into account information driven by literature of these 4 consensus molecular subtypes. MUST proposes several outputs, like the spatio-temporal evolution shown on the left. Here you can observe only a specific simulation, where the left panel shows the tissue evolution over time, and the 3 panels on the right from top to bottom report information about glucose, oxygen, and signaling molecules over time. In order to observe all the simulation at once, this is not a suited graph. So we need to switch to a temporal evolution graph. In particular, in the upper subgraph, the number of not completely tumor-free simulation in a determined instant is shown, while in the below subgraph is shown the cardinality of the agent counts across the non-completed tumor-free simulation in a log 10 scale fashion, and in particular the dotted line represents the average count of the agent in all the non-tumor-free simulation, and the shaded area represents its variability. In the real case scenario simulation, it is possible to observe clearly that CMS1 tends to be much more active from the immune point of view and much more infiltrated, as can be seen from the signaling molecule panel. CMS2 instead presents a low and constant proliferation, while the CNS3 that is consuming a lot of glucose, removing nutrients to other cells, it remains much more localized. And then, finally, in the CNS4, it is shown how a strong stromal invasion is able to promote the tumor growth. Then, in order to observe emerging properties of TME in different CNS settings, we simulate the effect of other parameters. And in particular, now we are seeing how the change in the glucose consumption from the default to up to 150-fold in the CNS4 produce different kind of simulations. And in particular, we note that as the demand for nutrient increases, we note that it is possible to observe that the tumor proliferation drops even when cancer-associated fibroblasts are recruited in the simulation. In conclusion, I have presented a hybrid multi-agent spatio-temporal model that can mimic spatio-temporal dynamics, model innate and adaptive immune system responses, can be informed from omics data, and can simulate colorectal cancer in agreement with the literature, even if only quantitatively. Nevertheless, we would like to find a novel way to inform what must, mainly through new bioinformatic analysis, like flux balance analysis, to better understand the nutrient consumptions, or leveraging on new data, like spatial transcriptomics, to better initialize our simulations. And we also want to further investigate patient-specific simulations and treatment effects. Thanks for your kind attention, and also thanks to all the organizers for this great opportunity, and I hope to see you at the conference in Lyon.